Welcome back, life science learners. In our segment, we're going to wrap up our understanding of cellular respiration. We're revising cellular respiration, and the best way to do that is by applying our understanding to solving a few questions. I encourage you to enjoy this lesson by interacting. As I go through these questions, I'll give you an opportunity to think about these answers, and if you can jot them down when we go through these, I hope that you can reflect on your answers and see if you were spot on. So let's attempt a few more questions on further application of cellular respiration. Question 4. A baker is testing a new recipe in order to bake the perfect loaf of bread. The two main ingredients in the bread dough are flour and yeast. She divides the dough into 3 equal parts and subjects all the loaves to different treatments before baking. Loaf A, she immediately bakes the bread. Loaf B, she covers the dough with a damp cloth and leaves it in a warm area for an hour before baking. Loaf C, she covers the dough with a damp cloth and leaves it in the fridge for an hour before baking. After baking the 3 loaves, she measured the height which each of the loaves had rose. The results are shown below. Loaf A, as indicated here, doesn't look like it has increased significantly in height. Loaf B has a bread that has risen very handsomely, and loaf C seems to be a moderate amount of height increase. So essentially this experiment is looking at the effect of different factors. And if you go back to the question, the different treatments are seen where the individual has kept them in a warm environment, then in a fridge, and then she's immediately used or placed them into the oven. So what's important in that, we've identified that there is the effect of different treatments on the baking of the bread. So she's mixed the dough with yeast and flour, and she's allowed that to stand for a little while in different temperatures. So let's formulate a hypothesis for this investigation. This is a 2-mark question. Guys, when putting together a hypothesis, it's important that we first link our hypothesis to the aim of the experiment. Remember that a hypothesis is often an educated guess that indicates what the possible results might be or anticipated results may be. It's important that when you string together the hypothesis that you include the independent variable, that is what is being tested, along with the dependent variable in terms of what is being measured to draw a conclusion. In this particular experiment, we know that the different treatments in terms of temperature is what is being tested. And the way in which we measure the effectiveness is by looking at the height, as she mentioned here, measure the height. So the height is being measured as an indication of how much the bread has risen. And that often is an indication of whether that method or treatment was effective. So a possible hypothesis would be bread dough kept at warm areas will rise the highest after baking. Or you can also say that the bread dough kept in the coldest area or the cooler area will rise the least. And so what we've identified is that we've identified the independent variable, which is the concept of temperature, as well as the dependent variable, which is the measurement of the height in terms of how much the bread rises. So that is a hypothesis. Again, it is a statement and not a question that of intent, where you say to investigate or to determine or to find out. All of those would be the aim of the experiment. Let's move on to question 4.2. The question requires us to identify A, the dependent variable. So I want you guys to think about that. So the dependent variable is that variable that is used to be able to draw a conclusion. So what was measured in this experiment that helped us to draw a conclusion about the effectiveness of different temperatures. I'll leave that to you to think about. The next part was identify the independent variable. And I often remind my learners to think about this question when trying to establish the independent variable. What is being tested in this experiment? We're testing the effect of something, some factor on the process of the bread dough rising. So think about that. And finally, one controlled variable. A controlled variable in this experiment would be something that is subjected, that you keep constant and subject all the different testing samples to. So a control variable is used to ensure that we have a fair test. So there was one variable that was kept constant in this experiment. So let's unpack those answers now. So the dependent variable in this case is the height of the bread. So in order for us to determine which was the most effective method of treatment, we measured the height of the bread. And so that was helping us to draw a conclusion, and we were able to say, well, that's probably the most effective method that was used to get the highest, or the loaf of bread to rise the highest. So the height of the bread is the dependent variable. The independent variable is the different temperatures. As we mentioned, the different treatments. In this case, there was immediately placed into the oven. In loaf B was placed in a warm environment for an hour, and loaf C was placed in a fridge. So it's the different treatments, and in this case, it is the different temperatures if you think about it as a variable. And finally, the control variable was the same amount of dough has been used. So it also mentions that the dough was divided into 3 equal parts, and this ensures that all the amount of dough was equal. So when we measure the height, we know that the height is in relation to the initial amount being the same. Which loaf do you consider to be the control for this experiment? Give a reason for your answer, and that's a 2-mark question. Guys, it's important that at this point we reflect on the difference between the control and a control variable. In any experiment, we have variables that we keep controlled, which all the test samples are subjected to, and that we refer to as a control variable. As you've just seen, we've spoke about the dough being divided equally, so the amount of dough was equal, and that's a control variable. However, this question requires us to analyze what was the control for this experiment. And guys, the best way to identify the control in any experiment is to ask yourself the following question. Which of the different samples were not subjected to the independent variable? Meaning, which of those different test tubes or samples, in this case loaves of bread, were not subjected to the different treatments? We know that the loaf of bread B was kept in a warm environment. Loaf of bread C was kept in a cooler environment in the fridge. Loaf A was placed immediately into the oven. So that was subjected to no specific treatment. So the rationale for saying that A was the control was that it was not exposed to any temperature, but baked immediately, meaning that it was not subjected to the independent variable of different treatment. And so it's important that you differentiate what a control is and what a controlled variable is. And so having a control in any experiment ensures that your test is reliable and it produces results that are accurate. Let's move on to question 4.4. One of the main ingredients is yeast. Name the function of yeast in this investigation. And that's a 1-mark question. So guys, the main ingredient having yeast in the experiment is to ensure that the process of cellular respiration takes place. During cellular respiration, we know that carbon dioxide is produced. When we bake bread, the carbon dioxide is released, and that is what causes the dough to swell up. And that is what makes the bread fluffy and moist. So when you cut through a loaf of bread, you often see pockets or little holes in it. And that is the air bubbles that are produced during the fermentation or during the yeast producing carbon dioxide. And those bubbles accumulate and cause the dough to swell up. And that's essentially the significance of having the carbon dioxide being produced by yeast. Which biochemical process allows the yeast cells to perform its required function? And so guys, we've been looking at cellular respiration. So it's a process of cellular respiration that is causing these bubbles to be produced. And finally, what is the yeast source of glucose? So the yeast cells require a source of glucose to be able to carry out cellular respiration. If we look at it, there is flour that is provided, and flour is a type of carbohydrate which is a complex form of glucose. So the glucose is going to be provided by the flour, which again contains carbohydrates. So the answers to these questions would be, again, the starch is present in the flour. We know that that process that we've just discussed was cellular respiration, and the process of using sugar is that the carbon dioxide causes the bread to rise. Right. What conclusion can be made from the results of the baked loaves? So guys, when we've looked at this, we measured the height of these loaves, and we can clearly see that the height of bread loaf B was significantly higher than loaves C and loaf A. So what is the conclusion? The conclusion needs to relate to the aim of the experiment. The aim of the experiment was to determine the effect of different temperatures. So let's link our aim to that. So it's got to link to the effect of the temperature or the different treatment. So this loaf B was subjected to 1 hour of being placed in a warm environment. And so when bread dough is left to rest in a warm environment, it will cause or result in bread rising significantly higher as compared to bread that is kept in a cooler environment. So I'll string that together in my answer here. So as I mentioned, the bread dough that must be kept in a warm area for a period of time before baking so that the bread can rise. Okay. And finally, how can the bread maker improve the reliability of her experiment? Guys, it's important that we're able to reflect on any experiment and comment on the accuracy of our results as well as the reliability. In this particular experiment, the baker had separated the dough into 3 different parts. All she did was use one dough for each of the different temperatures. In order to improve the reliability of her results, it would mean that she could repeat the experiment and see if she got the same results, or she could increase the amount of dough and have 2 lots of dough for sample A, sample B, and sample C. And that would mean that you would be able to see if those results are consistent. So guys, we've looked at the application of cellular respiration, we've unpacked the different stages, and we've looked at how it's used in industry. I trust that you've enjoyed the lesson, and I hope that you can go back, reflect on what we've just done. From my side, cheers, have a bio-lekker day.